Before developing into an adult organism, every living being is an embryo at its initial stage of life. Do you know how an embryo develops into an adult organism? It is through the development and the differentiation of cells. The differentiation of cells is a result of coordinated gene expressions. So, what is a gene expression? Or how can you define a gene expression? Gene expression is one of the mechanisms used by the cell to regulate the production of gene products such as RNA or proteins. There are several factors that regulate gene expression, and they are metabolic, physiological, and environmental conditions. An example of the regulation of gene expression is the synthesis of the sugars with the help of beta-galactosidase enzyme by E. coli. The beta-galactosidase enzyme is the enzyme that hydrolyzes lactose. What happens when there is no glucose in the cell environment? In the absence of glucose, the enzyme hydrolyzes lactose into galactose and glucose, and the E. coli derives energy from the obtained glucose. So this is a process of hydrolysis is an example of regulated gene expression. Through hydrolysis, E. coli derives energy. Now, let us see how the regulation of gene expression occurs. The regulation of gene expression differs in prokaryotes and eukaryotes. In eukaryotes, regulation could be exerted at several stages. The stages in eukaryotes where regulation can be exerted are transcription level, processing level, transport of mRNA, and translational level. At the transcription level, regulation of gene expression takes place during the formation of the primary transcript. At the processing level, the regulation would be in splicing. The other level where the regulation occurs would be during the transport of the mRNA from the nucleus to the cytoplasm, where the splicing, capping, synthesis, and tailing take place. At the transitional level, regulation of gene expression takes place by controlling the levels of protein synthesized by the mRNA. So that was all about the transcription in eukaryotes. In prokaryotes, the predominant site for the control of gene expression is a transcriptional initiation site. Let us see how the regulation happens at the initiation site in prokaryotic transcription. The process of gene expression is somehow regulated. Accessory proteins play a major role in regulating the gene expression. And these accessory proteins act positively and negatively. Do you know how? If the accessory proteins act as activators of the process, it infers that if they increase the rate of transcription, they are acting positively. While on the flip side, they act as inhibitors or negatively, that is, they decrease the rate of transcription. Let us check the functioning of accessory proteins. These proteins regulate the activity of the enzyme RNA polymerase in the promoter region. Have you ever thought of the output of these regulations? This regulation affects the ability of the RNA polymerase to recognize the start sites. The accessibility of the promoter region is regulated by these sequence proteins known as operators. So, where can you find these operator genes? These operator genes are seen near to the promoter and the repressor gene in operons. Now, another question may arise in your mind. What is an operon? We can define an operon as a genetic regulatory system in prokaryotes consisting of multiple genes with a promoter and an operator site. For example, there is a specific operator present in the Lac Operon and a specific repressor. The term polycistronic implies one gene which codes for more than one polypeptide. The E. coli Lac Operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugar used for cellular metabolism. This is also known as the Jacob and monads concept. Now, let us check the unit of a Lac Operon. Lac refers to the lactose in the Lac Operon. The unit comprises three structural genes, that is Z, Y and A. The Z gene codes for beta-galactosidase, which catalyzes the hydrolysis of lactose into glucose and galactose. Y gene, which codes for permease, which regulates the lactose permeability in the cell. And A gene codes for trans-acetylase and assists the enzyme beta-galactosidase. One promoter gene, that is a P gene, one operator gene, that is the O gene, and one regulatory gene, which is the I gene, which codes for the repressor of the Lac Operon, is found upstream to the promoter that codes for Y, Z and A genes. The presence of an inducer inactivates the repressor. The inducer can be of lactose or allolactose. The inactivation takes place through the interaction with the inducer. The lactose is available in the growth medium. The lactose is transported to the cell with the help of the permease enzyme. So you need to note that a very low level of energy will reside every time. Here, the mechanism is that in the absence of lactose, the repressor of the operator is synthesized from the I gene. The repressor protein binds to the operator region of the Lac Operon and prevents the RNA polymerase from transcribing the operon. And this regulation of Lac Operon by a repressor is referred to as negative regulation. If lactose or the allolactose is present, then the repressor gene is inactivated by the inducer, thus allowing the RNA polymerase to get bound to the promoter and the transcription occurs. Proteins will be synthesized by the translation from each of the structural genes, as these proteins are responsible for the metabolism of lactose. This regulation of Lac Operon by a repressor is referred to as positive regulation. We now know about the regulation of gene expression in prokaryotes and eukaryotes. An example of such regulation of gene expression is the Lac Operon. Let's summarize what we have learned.
Manorama Horizon. Learning made simple.